Thank you for inviting me to present the results of our STOP 101 study at this conference. My name is Dr. Stephen Shrewsbury, and I am the Chief Medical Officer for Impel Neuro Pharma, who conducted this three-way, three-arm, six-sequence study in healthy adult volunteers to compare the bioavailability and pharmacokinetics of our product, IMP-104, delivering 1.45 milligrams of dihydroergotamine mesylate, or DHE, to the upper nasal space, to the approved traditional lower nasal space spray delivering 2 milligrams, and to the approved 1 milligram intravenous DHE product. The study objectives, in addition to comparing bioavailability, were to also assess safety and tolerability of single doses of the 3 DHE products to healthy adult volunteers. The IMP-104 product consists of a single-use precision olfactory delivery device and an amber glass vial of dihydroergotamine mesylate solution. Preparation for use consists of taking the top off the vial and removing the plastic protector off the bottom of the device, screwing the vial onto the device, and spraying the device 4 times to prime it. Dose administration is 2 sprays, 1 to each nostril, and the assembled device and empty glass vial are then disposed of. Traditional nasal sprays, such as the comparison product Migranal used in STOP 101, deliver a cloud of liquid droplets to the anterior and lower nasal space, as shown in this model of a nose using blue dye. The droplets coalesce on contact with the surface, and then, often, liquid may run out of the nostril onto the upper lip or down the back of the nasopharynx. In contrast, the pod system uses a propellant to carry and then push in a single smooth actuation the drug product through the nasal valve and into the highly vascular upper nasal space. Each actuation takes just 0.3 of a second. The actual drug product formulation is identical between Migranal and IMP-104. However, IMP-104 is delivered as 2 sprays carrying 1.45 milligrams of DHE delivered sequentially. Migranal is delivered as 2 milligrams, requiring 2 sprays, a 15-minute pause, and then another 2 sprays. The STOP 101 study was designed to assess if delivering a lower dose of DHE could provide a similar or better rate and extent of DHE absorption to the approved nasal spray, and how it would compare with the IV D.H.E. that many headache centers use as their go-to treatment for severe migraines, status migrainosus, and other headache conditions. The phase 1 study was conducted in the 4th quarter of 2017 in an experienced clinical research unit in Australia, but after FDA had provided input to the protocol, specifically requiring that we tested 6 different treatment sequences in the 3-way, 3-arm, 3-period crossover study to mitigate the risks of a carryover effect from one treatment to the next. After obtaining consent and an initial screening, eligible subjects were randomly allocated to one of the 6 treatment sequences and were domiciled in the clinical research unit for each of the 3 48-hour periods of PK blood draws, which were more intense in the 15 hours following each dosing, and during which standard safety assessments were focused, with 7 days separating each dosing. The safety assessments consisted of collection of adverse events, vital signs, ECGs, and blood and urine collection for hematology, clinical chemistry, and urine analysis. Slide 7 shows the disposition of subjects in each of the 6 balanced sequences that entered and completed the 3 treatment periods, and the reasons for any discontinuations. We ended up with 28 subjects completing the study, providing 27 sets of pharmacokinetic data from the 38 healthy volunteers who enrolled. Slide 8 shows the demographics for the 38 volunteers who enrolled in each of the 6 treatment sequences, with the overall demographic data showing the study recruited mostly young adults, mean age 30 years, with a good gender balance, 52% male, mean healthy adult body weight of 71 kilograms, and non-obese mean body mass index of 23 kilograms per meter squared. The youngest subject was aged 21 and the oldest was aged 47 years. Treatment emergent adverse events were reported by more subjects after the slow bolus IV dose given over 1 minute, 66%, than with either of the nasal delivered doses of DHE, which were reported by 48% for IMP-104 and 41% for the DHE nasal spray. The most frequently reported treatment emergent adverse event was somnolence in these healthy volunteers at rates between 13 to 28% across the 3 treatment cohorts, with headache reported by 7 to 19% and dizziness, notably almost exclusively with the IV product at 16%, and nausea at 9% after IV D.H.E, despite pre-treatment with metoclopramide. Both nasal doses of DHE were pre-treated with metoclopramide, which may explain why only one subject reported nausea with either nasal product. The IV product was also associated with cases of restlessness, 4 cases, hot flush, 3 cases. Overall, both nasal delivery systems were well tolerated and few adverse events after these 2 treatments were blamed on the investigational product. No treatment-related serious adverse events were recorded. On this graph, note that the vertical axis is plotted as a logarithmic scale. When injected intravenously as a slow IV bolus over 1 minute, 1 milligram of DHE has, as expected, a high and early maximum concentration, or Cmax. This is responsible for the nausea that is frequently reported with IV administration, and the reason the ethics committee required us to pre-treat subjects with metoclopramide. In contrast, delivering 2 milligrams of DHE through a traditional nasal spray as 2 sprays, and after a 15-minute pause, a second pair of sprays, sees a slow rise in blood levels of DHE to reach Cmax by approximately 1 hour after dosing. As it can also be seen, the wide error bars show that the nasal spray demonstrated wide variation in the blood levels after dosing, even in the controlled environment of a clinical research unit. In contrast, the blood levels obtained after delivering the exact same formulation of DHE, as in Migranal, as just 2 sequential sprays, but 2 to 3 centimeters deeper into the upper nasal space with the pod device, generated a significantly different profile, seen here in red, closely approximating IV levels by 20 minutes and actually meeting IV levels and achieving Cmax by 30 minutes. The blood levels after IMP-104, 1.45 milligrams, then followed blood levels seen after 1 milligram of IV DHE all the way out to 48 hours, and with much less variability than after the traditional nasal spray delivery of 2 milligrams, which never reached IV levels. In fact, using the pod to deliver less than 75% of the dose of the nasal spray, we achieved 4 times the Cmax and 3 times the area under curve compared to Migranal. It has been postulated that the area under curve over the first 2 hours is the most important predictor of DHE efficacy in treating acute migraine. And the profile generated by IMP-104 has a very similar PK profile to a product that was developed about 15 years ago, MAP-0004, but never approved. That product was a suspension of DHE in HFA propellant delivered by a breath-actuated metered dose inhaler that was designed to deliver the DHE formulation to the deep lung. The program had a successful phase 1 PK study demonstrating a very similar PK profile to our current IMP-104 product. And then statistically significant positive efficacy results from 2 double-blind placebo-controlled studies, one phase 2 and a large phase 3 clinical study. Unfortunately, the product was never approved because of chemistry and manufacturing issues, likely lack of stability of the DHE in the HFA propellant over time. At Impel, we specifically set out to try and replicate the PK curve that MAP had generated, knowing that the product had such good efficacy in the clinical program. However, in all the Impel pod programs, the HFA propellant and the drug product do not come into contact until the point of delivery, so avoiding the stability issues that MAP had encountered. In addition to establishing safety and efficacy through comparison to 2 marketed products, these Stop 101 results are also supported by the hypothesis developed by MAP that the area under curve over the first 2 hours is the most important predictor of effectiveness. As can be seen in this table, that not only does IMP-104 generate the 4-fold higher Cmax for less than 75% of the dose of the nasal spray, and 3 times the area under curve over time to infinity, but the coefficient of variation is less for the key pharmacokinetic parameters. Importantly, the Cmax, however, is 1/10 of that seen after IV slow bolus injection over 1 minute. In summary, the Stop 101 study showed IMP-104 was well tolerated in healthy volunteers, better than the IV D.H.E product, and with a similar rate of treatment-emergent adverse events as the approved nasal spray. Although delivering less than 75% of the dose, the pod system enables the DHE formulation to be absorbed more rapidly and extensively via the upper nasal space, leading to a 4-fold increase in Cmax and a 3-fold increase in area under curve, with a particularly encouraging profile over the first 2 hours. The Cmax is 1/10 of that of the IV DHE, but blood levels approach those after the IV administration by 20 minutes and match them from 30 minutes to 48 hours with greater consistency than the traditional nasal spray. When asked, these mostly nasal delivery naive volunteers stated they preferred the pod delivery system to the traditional nasal spray in 69% of cases at completion of the study. The bioavailability of IMP-104 at almost 60% suggests the upper nasal space may be an efficient route of delivery of at least some drugs, and in this study was better than when the same formulation was delivered to the lower nasal space, providing only 15% bioavailability. The lower coefficients of variation showed more consistent key pharmacokinetic parameters for IMP-104 compared to the nasal spray, suggesting more reliable and consistent delivery and absorption despite the reduced dose. The study was well powered, greater than 99%, to show the study met the statistical requirements that were set. These pharmacokinetic benefits should translate into a better efficacy profile for IMP-104 in the clinic, with similar safety and tolerability to the approved nasal spray, and the study data will be pivotal to the IMP-104 new drug application. The PK data will act as a bridge to the established efficacy data of the approved nasal spray and the established safety of the IV product, and allows the new drug application to be completed by the collection of safety data over 24 or 52 weeks. The Stop 301 study was discussed with the FDA ahead of its execution to provide this data. The Stop 301 trial was a safety, tolerability, patient acceptability, and exploratory efficacy study of IMP-104, conducted at 38 research sites in the US. It initiated in June 2018 and completed in March 2020. The study had a 4-week screening period using best usual care, a 24-week treatment period for all patients, and a 28-week extension for a subset who continued to suffer a predefined minimum number of 2 migraine attacks per month. A daily e-diary collected daily data and migraine attack information throughout. After the screening period, those who had had at least 2 migraine attacks were offered open-label IMP-104. The primary objective of this study was to assess safety and tolerability of intermittent DHE delivery to the upper nasal space using upper nasal endoscopy, which was performed by qualified and experienced otolaryngologists, and University of Pennsylvania smell identification tests, or UPSIT, to assess olfactory epithelial integrity and olfactory function, respectively. In addition to standard adverse event reporting, vital signs, ECG and laboratory tests at pre-specified visits. Exploratory endpoints included efficacy collected on the e-diary as pain and most bothersome symptom freedom and relief in the 48 hours post each dosing, with a focus on the first 2 hours. The diary also collected data on the use of other medications. In addition, patient acceptability was collected when patients completed their treatment periods, either at 24 or 52 weeks. 2 patient-reported outcomes were also collected: the Migraine Disability Assessment Test, or MIDAS, and the 6-question Headache Impact Test, or HIT6. And healthcare resource utilization was reported for the 12 months before enrolling and during the 24 and 52 weeks on study. 360 patients entered the initial 24-week period, of which 74% completed this period, and 73 went on into the 28-week extension, of which 90% completed 52 weeks. Patients were given printed instructions for use that had been developed through a comprehensive human factor program and enough IMP-104 single-use product to dose a maximum of twice in any 24-hour period, a maximum of 3 times in any 7-day period, and a maximum of 12 times in any 4-week period, similar to established products. We were fortunate to complete the study as the current COVID-19 pandemic was kicking off, and although site staff were being furloughed and the last few visits were challenging to schedule and complete, site closeout visits and the process of wrapping up the study were both completed on schedule in the first few months of 2020. Several populations were identified in the study. All patients entering a treatment period were designated as the full safety set, while those completing the 24 and 52-week periods and who had continued to report a minimum of 2 migraine attacks per month were designated as the primary safety set, as that was the population the FDA wanted to review for safety assessment. I shall concentrate on presenting data from the full safety sets in both periods, but the data is very similar for the primary safety sets. Of the 360 patients who enrolled, 354 reported at least one self-recognized migraine in the initial 24-week period. 262 of those patients, i.e. 74%, completed 24 weeks of treatment, despite the burden of procedures, especially nasal endoscopies at otolaryngology clinics and collecting daily diary data, which did account for 25 withdrawals by subject. Other reasons for withdrawal were lack of efficacy in 21, 6%, adverse event 24, 7%, loss to follow-up 11, 3%, non-compliance with the protocol in 5 subjects, and physician decision in 1 case. The baseline demographics were fairly typical for a migraine study, with mean age of 41 years, 86% female, suffering from a mean of 4.6 migraines per month in the baseline period. When asked at baseline to identify their most bothersome symptom, approximately 50% stated photophobia, with approximately 15% reporting phonophobia or nausea. However, over 5% reported foggy thinking as their most bothersome symptom, which may be under-recognized and underreported in migraine studies. Other most bothersome symptoms included vomiting in 3%, visual change in 3%, fatigue in 2%, dizziness and vertigo in 1%, sensitivity to touch in 1%, with various other symptoms reported in 22 patients or 6%. The mean duration from time of migraine diagnosis to enrollment in this study was 19 years, and all patients had over the years discovered what available medications worked best for them in relieving their migraine attack. Sometimes a combination of products, and this was noted in their 28-day baseline period and was referred to as their best usual care. Note 28%, 28% used a triptan at baseline, 44% used acetaminophen, and 38% took a non-steroidal anti-inflammatory drug. Patients could use more than one class of drug, and often did, and many, 16%, took combinations of products. Slide 18 shows the treatment-emergent adverse events that were reported over the 2 treatment periods by the respective full safety sets. In the first 24 weeks, nearly 5,100 doses of IMP-104 were self-administered to a self-recognized 4,515 migraine attacks. Over this 24-week period, just over a third of patients, 37%, reported a treatment-related adverse event. And over the full 52 weeks, under half of patients, 45%, reported at least one event. The vast majority, 96% of these treatment-emergent adverse events over the 24 weeks were mild or moderate in intensity. The most frequent was, not unexpectedly, nasal congestion, by 15% and 18% in the 24-week and 52-week periods respectively, which rarely, however, caused a subject to discontinue taking IMP-104. Encouragingly, nausea, often reported by approved DHE products, was only reported by under 7% of patients. And unlike the STOP 101 study, this was in the absence of any pre-medication with an antiemetic. No deaths or treatment-related serious adverse events were reported during the full 52 weeks of the STOP 301 study. No cardiac events were reported throughout the study, but patients with a history of coronary vascular or cardiac disorders were excluded from participation, consistent with the labels for approved DHE products. In addition, nasal endoscopy findings were scored by the examining endoscopist and UPSIT test scores, and both of these were reviewed by an independent panel of expert otolaryngologists who found no new safety findings of concern compared to existing nasal spray, and stated their opinion that the reporting of treatment-emergent adverse events was sufficient for monitoring of upper nasal safety in both future clinical research and potential approval and clinical use. Patient-reported exploratory efficacy compared the use of IMP-104 on study to the results seen with their best usual care during the 28-day baseline period. The recommended co-primary endpoints for migraine studies of pain freedom and most bothersome symptom freedom at 2 hours both increased, from 30 to 38% for 2-hour pain freedom, and from 46 to 52% for most bothersome symptom freedom at 2 hours. IMP-104 effect, as measured by pain relief, was reported to start as early as 15 minutes after dosing, rising to a pain relief rate of 66% by 2 hours. Not only with high rates of sustained pain freedom over 24 hours of 93% and over 48 hours of 86%. Not only did 74% of patients persist throughout the 24 weeks, and of those completing the 52-week period, over 90% persisted, but 84% of patients who did complete and answered the patient acceptability questionnaire reported IMP-104 was easy to use, and most patients preferred IMP-104 over their best usual care. Lastly, the benefit from IMP-104 was maintained even if the migraine attack had been underway for over 4 hours by the time of dosing. Thus, in conclusion, IMP-104 was well tolerated when self-administered in this 52-week open-label study with no new safety concerns compared to previously approved nasal DHE products. Patient acceptability was very encouraging, with the majority finding it easy to use and preferred to their current treatment. Although this was an open-label study, patient-reported exploratory efficacy showed higher rates of pain freedom and most bothersome symptom freedom at 2 hours than they achieved with their best usual care in the 28-day baseline period, with relief being experienced as early as 15 minutes and very high rates, 98%, of sustained pain freedom at weeks 21 to 24. Data from the STOP 301 study suggested that IMP-104 may provide rapid, effective, consistent, and well-tolerated alternative to IV D.H.E, and with the added advantage that it may be self-administered outside of hospital, infusion centers, and headache clinics, where IV D.H.E remains a popular option, especially for headache experts. The results from the STOP 101 and STOP 301 trials have now been submitted to the FDA as a new drug application, and we look forward to working with the FDA towards the goal of bringing IMP-104 to the US market as a self-administered, easily delivered DHE product available to patients suffering from an acute migraine attack with or without aura. Thank you for your attention.